Hi everyone, this is Mr. Sinti, and this particular video is on the concept of osmosis. And I think the name osmosis precedes this conversation because I'm sure you've heard of it before somewhere in your past, and if that's the case, I think it sort of signifies its importance. And so let's just jump right into this. And what I want to say about osmosis is this. If you have a cell like this, and you had water on the outside of the cell. Let's make water blue. And you said water is on the outside of the cell and water is on the inside of the cell. You have to ask yourself the question, is water capable of passing through the cell membrane, first off? In other words, is water permeable to the cell membrane? And the answer to that is yes, of course. Water can simply move in or out of the cell. And it moves because the molecules are constantly in motion. So the question becomes, is there more water moving in or is there more water moving out? And the answer to that question is fairly simple. In other words, if I were to create another cell and if I said something to you like, would water, if it was high concentrated on the outside of the cell, move to the inside where it's lower? Will water move in or out of the cell? And of course, being great students that you are, would say water obviously would move from an area of high concentration to an area of low concentration. So if water goes from an area of high concentration to low concentration across a cell membrane, dun 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 dun, this is what we call osmosis. And I know what you're thinking, wow, that's kind of a letdown, because all it really is the diffusion of water. And in fact, that is all osmosis is, the diffusion of water. But there's some other considerations that I want to factor into this conversation. It is the diffusion of water, but let's consider a few interesting concepts. Like, for example, let's go back over here and say, well, you can ask yourself the question, here's another cell, why did water move into the cell? And you're like, I know, because it was in high concentration. Well, when we're talking about water, we're not necessarily talking about the quantity of water that we do when we're talking about other solutes. When we're talking about water, there's another factor at play here, solutes. And so if I said that there were a lot of solutes, this could be anything. This could be sugar or salt. Can you see how if I put a lot more solutes on one side of the membrane versus the other? Let me introduce a term that's really important here in this conversation. If I say an area has a lot of solute, it's considered to be, this is now the inside of the cell, considered to be hypertonic. Hypertonic, and hyper meaning high, like a lot of sugar. And then if this is very hypertonic on the inside, hypertonic, then this outside part of the cell, this outer part of the cell is considered to be hypotonic, meaning low. Like this prefix, not a word expert like others, but this prefix means below. Like, for example, if you're getting a shot in your skin, it's used by a hypodermic needle, which means below the skin, or hypoglycemic means low sugar level. So this is hypo-environment. And so what does that have to do with the diffusion of water or osmosis? Well, let's consider the effects of hypertonic and hypotonic on osmosis. Let's do that. So what I want to say, this is from our class notes, just want to establish these terms first off. If a solution has a high concentration of solutes, as I mentioned, it's considered to be hypertonic. If the concentration of solutes are low, it's considered to be hypo. And if the solution has equal amounts, let's try that. Let's make a cell and we'll make 3 X's on the inside and 3 X's on the outside. Then it's considered to be isotonic, meaning the same concentration. Iso meaning the same. So, okay, you're saying to yourself, what does this have to do with the movement of water? Well, what happens is, if you can imagine sugar being on one side of a cell membrane and sugar on the other side, the sugar is going to interact with the water molecule, and it's going to prevent water from moving from one area to the other. Now, although that's clear in words, I'm not sure if it's clearly understood until you actually see an example of this in an illustration. Say I had a glass tube that was shaped like a U, and say it's a U tube. Pun intended. So it's a U tube, and you had water in the U tube, and it was separated by an artificial membrane. See this dotted line? Let's say that that's a membrane, and it's permeable. And it's like, okay, it's permeable to what? It's not permeable to these dots, because as you can see, these dots are too big to pass through. But it is permeable for water, so water can move across the cell membrane. So if I ask you the question, where will water move? And let me label this side as being side A of the U-tube, and this is side B of the U-tube. One of the things that we need to consider is the simple idea of osmosis. Water will go from high concentration to an area of low concentration. And this is osmosis across the cell membrane. And you're like, okay, well, where's water going to go in this diagram? Well, water's going to move in response to solute. Like, for example, do you see on this side there's a lot of solute? If the area has a lot of solute, remember this term, we call it hypertonic, meaning lots of solute. And then over here, there's not a lot, so it's considered to be hypotonic. You're like, well, what is the effect of this? Okay, let's consider this. What is the effect of this? Let's come over here. I want to show you this little animation. This one right here. Okay, let me let this play and tell you what, and sort of narrate this. Do you see how here's our U-tube, and here's the cell membrane, and there's water. Now remember, water's in constant motion, so let's consider this. So the water's going back and forth like this. But then, if I were to add a solute, if I were to add salt, let's say over to this side, I would create what's known as a hypertonic environment. In other words, more salt on this side, not a lot of salt on this side. This side would be hypo. So let me add the salt. And when I add the salt, I want you to watch specifically what the water is doing. Do you notice in the beginning the water is going back and forth evenly, back and forth? But when I add the salt, check this out. I add the salt, and notice what the water's doing. Do you notice how the water is clinging right here to the salt? Because when salt goes into the water, it breaks up into sodium and chlorine ions. Notice how the water is attracted, because water's polar. And if water's attracted to these ions, what are the consequences of that? What's interesting is this. This is the main point in osmosis, and I think most people don't appreciate this subtlety, is that when water is interacting with the ion, it's less likely to move to the other side. Water moves to the left, water moves to the right. Water moves to the left, water moves to the right. But it's kind of like a party's going on in here. Like you put salt or you put sugar in on this side, and what's happening is, because this is hypertonic, the water is engaging. It's sort of like when your parent comes to pick you up at the party, you're not wanting to leave. So water is interacting with these solutes, so it's less likely to move in the other direction. And if it's less likely to move into this direction, more water will move over to where the salt is. Do you see the subtle difference with that? Let me continue with this animation. Okay, this is an important point. So in this animation, you can see here, this is some sort of like gas, like carbon dioxide or oxygen. You can see how the membrane is permeable and these molecules are moving across. Now, if you change the scenario up and you say, okay, now here's these big yellow balls. These ones are some kind of ion, they can't move. But water is small enough to move across the cell membrane. So here's our scenario. Let me pause it. Do you notice here how water is moving equally? Left and right, because water can move. So there it is. It's moving left and right, left and right. So diffusion of water is equal. Now, so now I've added a solute to one side. I'm not sure if you've heard of this before, but this particular solute is called urea. It's actually a nitrogen compound that's polar, and we actually produce it in our urine, and this is what we urinate out. It's a nitrogenous waste. So if I add urea to this side of the beaker, this side is considered to be now hypertonic because it has a lot of solute, and this side over here is relatively solute free or hypotonic. Now, where's the water going to go? And you're like, oh, the water's going to move from an area of high concentration to low concentration, but watch the video closely. So, do you notice here how the water is starting to interact with the urea? It's less likely to be moving across the cell membrane like it was before. So you notice how it's interacting? So all things being equal, some water will go left, some will go right, but because of this interaction of solute, more water will be going from the left side to the right side. So more water will be going like into the party, and so the water level will go up. And so what's interesting is students like to sort of like, give me something to memorize, and I've never really liked the idea of memorizing, but water moves from an area of hypotonic to an area of hypertonic environments. Water goes from high concentration to low concentration, but what's lowering the concentration of water is the solute present. And so this is a very interesting point. So if you had equal solute on both sides, do you remember what that was called? If it was equal solute on both sides, that's considered to be isotonic. And you're like, well, now what's going to go on? Well, a little bit of water this way, a little bit of water that way, and so there'll be no net gain. And so there would be no movement of water at all in an isotonic environment. It only moves when there's an unequal amount of solute. And so this is a very important concept in biology, needless to say, although I will say it. And so osmosis is the movement. As you can see, the water goes over in this direction. Let's play this out. So a cell's survival depends on the uptake and loss of water. It's important that cells don't lose too much water or gain too much water because it's harmful both ways. An equal amount of water movement is more stable. Let's consider an example of this. Like, how about our own red blood cells? Like, before I show this diagram, let me go and draw my own here. Let's go up here and say I had a beaker of water. And you're like, okay, a beaker of water. It's very windy. And so the water, let's say it's distilled water. Did you know distilled water has no solute at all? There's nothing in it, and so it's the most hypotonic water that you can have. Say I were to prick my skin and I get some blood and I dropped in a red blood cell, just one cell in a glass of distilled water. Let me label that distilled water so you can remember it. So distilled water. So there's no solute. Now, can you appreciate that inside of a red blood cell that there's lots of iron called hemoglobin, protein, hemoglobin, and there's a lot of salt and there's a lot of sugar in there, so there's a lot of solute. Would you agree that this is the more hypertonic environment and then out here is hypo? So if I asked you, where's the water going to go? You would say, well, it's going to go into the cell. It goes from where it is to where it isn't so much, and so the water goes in, and you're like, oh, what's the big deal? What's the point of this? Well, if water keeps pouring into a red blood cell, eventually it's not going to be able to handle that. And what's going to happen? I'm sure if I haven't never used the eraser tool. What's going to happen is so much water is going to build up in here that the whole cell is going to do what? It's going to rupture and it's going to explode, and the cell's going to pop. This is not good. And so let's go back here to our original cell notes here. Do you notice that if you put red blood cells in a hypotonic environment, that's what I was just describing, water's going to go in and it's going to cause the cells to like fill up like a water balloon, and it's not going to be able to handle that because the cell membrane, if you remember, is just a little phospholipid bilayer that's kind of oily. It's going to fill up and it's going to blow up and the cells are going to rupture. This is not good. This is not good at all. This is more like what happens. Our red blood cells that are circulating in our plasma, in our blood, the water, is in an isotonic environment. So water goes in, water goes out. If you were to take cells and put it in a real salty environment, In other words, I can make a beaker and fill it up with salt and then throw my red blood cells in there. Well, the cells are going to lose water and they're going to shrivel. This kind of is what is happening when, for example, when you're eating a little bit too much salty food. If you're eating salty food, your cells are pretty much losing water, and you become a little bit dehydrated. It also explains the reason that, for example, if you're out on a boat and you're thirsty, say you're sailing along the bay and you're like, well, I'm really thirsty. I think I'm going to just reach over and drink some bay water. I wouldn't recommend it because bay water, as you all know, is extremely wet. It's extremely salty. And so this is the situation. If it's very salty on the outside, like this, the outside is considered to be very hypertonic, right? And so what's going to happen is the water is going to go in to that environment. The water is going to leave. Do you remember, like, water is capable of going in, but it's more likely to leave because it goes from a hypo environment to a hyper environment. So what would happen is if you were to drink the bay, you'd become dehydrated by doing that. Don't do that. So as you can see here, I like this slide because it sort of contrasts what would happen in a red blood cell. That's what we've been talking about. You put it in distilled water, water goes in, it pops. Isotonic, it's great, in and out, in and out. If you put it in a salty or sugary environment, water will leave and the cell will shrivel. This is not good. But now something different happens in a plant cell. Do you notice how a plant cell has, let me emphasize this, a plant cell has a rigid cell wall. If you remember from our discussions in class, a cell wall is made up of a sugar called cellulose. This is pretty hard substance. This is like what wood is made out of. That's not really moving anywhere. The cell membrane is inside of the cell wall. Now what's interesting is if you put a plant cell in a hypotonic environment, in other words, if you were to put a plant cell in a glass of distilled water, you wouldn't have to worry too much because water would go in, and it would swell up, but the cell would never burst like a red blood cell because the cell wall is preventing it from bursting. And in fact, this is what keeps plants upright. This is what keeps them reaching for the sky in order to photosynthesize. This is why we like to put our carrots into a Ziploc bag with water so that the water goes in and so they're crispy. This is why we put our vegetables, for example, in the crisper drawer of the refrigerator because it's a high humid environment. Water keeps going in, and so when we pull our zucchini out of the refrigerator, it's nice and crisp, and we like that. What's interesting is isotonic environment actually puts a bit of a strain on the plant. It doesn't really like that. It actually prefers a hypotonic environment. And then this is maybe the worst for a plant. If you put a plant, just like an animal, in a hypertonic environment, instead of pouring regular water into a plant, if you poured salt water into a plant, like say you wanted to destroy your neighbor's garden, you can simply go outside and pour at night. You can pour salt water on their garden, and what would happen? All of the water inside the cells would leave, and the cells would eventually, their cell membrane you can see is pinching in. They would lose their pressure, and the whole plant would wilt and end up dying. It's kind of an evil thought. Sorry about bringing it up. But when a plant cell is in plain water, it's considered to be turgid, and that's a healthy state for most plants. They like it kind of crispy, and so that's very important. And so if it's in an isotonic environment, it's said to be flaccid or sort of steady. Plant may even wilt under those conditions. And then finally, if it's in a real salty environment, the term that describes this is called plasmolysis. That's the big word for the day, meaning that hypertonic environment, water leaves, the cell membrane pinches in, there's no more pressure, there's no more turgor pressure. If there's no more pressure on the cell wall, the whole plant starts to wilt, and then you see it, you say, I think the plant needs water, and then you water it. So I hope you enjoyed our conversation on osmosis. Thanks for watching.